Hello. Uh, so this is the first recording about this particular topic. Our idea here is to start addressing this particular tool, hidden Markov models to analyze sequence similarity. Uh, Nevertheless, in this video, we are not addressing that topic yet, but the main goal is to reach that topic at some point. So this first video is a very short video. The idea is to introduce a little about this idea of similarity to understand what is homologous sequences and provide some context about that. Okay, so the idea is here to start elaborating some material based on that we are working in this in the MEM lab as a group. So people that are arriving this semester, in that week, they can see some material and they can check some material and ask for questions or propose new topics about that. And at the same time, people that are already in the research group, they can also share some material or elaborate some material for older people or new generation of people. Yeah. So today I'm going to elaborate this short video. It will be very, very short, I promise. At the same time, to try to get some feedback to see if it is clear the way that I'm elaborating this video. Otherwise, if it is not clear, I can start addressing some points that are unclear in the second video. Yeah. So pretty much I'm going to start introducing the topic, and we are going to talk about homology. The idea behind homology is to understand if there is evolutionary ancestry in some sequences. We are analyzing here some sequences, maybe from amino acid to understand the proteins, or DNA sequences that will have some nucleotides there. Okay, the idea of infer homology, we infer homology when two sequences of a structure share some similarity. And what I'm saying, some similarity, what it means that we will have more similarity that will be expected by chance. Okay, so here I'm going to highlight some things, in particular, the idea here of inference. I would like to see if everybody's clear on this. Inference is to try to conclude or to draw a conclusion. We want to conclude homology, and this is draw a conclusion based on what? Based on evidence. In this case, provided by the sequences. Yeah, those sequences will be our data. So based on sequences and evidence and some reasoning. This is the idea of making an inference. Okay. And after that, as you may guess, well, maybe yes, maybe not. I'm going to start talking about some cutoff or some idea of similarities to try to understand what is this idea of more similarity that will be expected by chance. And for sure, well, I don't know if that connect with you, with your minds in the concept of statistics and probability. So this is what is behind. Yeah. And of course, the hidden Markov model uses some more, not necessarily complicated, but we need prior understanding of some basic things about statistics to get the idea of these hidden Markov models. Okay, so those are the two things that I would like to start talking about. What is the idea of concluding or of making a conclusion of what is homologous proteins or species or something like that based on sequences, and what kind of statistics we would like to use to try to make that conclusion. Okay. Also, I'm going to elaborate on what is the limitations that we can find here. Okay. So pretty much the motivation is this one. The motivation is behind what we know as structural biology. In structural biology, you can understand these organisms that share some similarity in the structure or the positions. In this case, you can see that they are comparing human, cat, whale, and a bat. And you can see something like a similar structure here. And the idea behind the similarity is illustrated using some colors here. Yeah. So for instance, you have one large bone that is purple, uh, and there are other modification other steps. So the idea of having a common ancestor in structural biology comes from this idea of similarity of a structure or position. Yeah. So we would like to try to understand the same on molecular biology, but not necessarily is always like that. Yeah. So this is exactly one of the limitations, but this is the main motivation to try to understand evolution based on similarity. Yeah. And for that, when you are analyzing sequences, what you do is, if for some reason you can observe some excess of similarity, the simple explanation of that excess will be that these two sequences uh, comes from one common ancestor. Okay, this is something like a simple explanation about that. Okay, simple is not necessarily means that is the right one. Yeah, so this is the simple explanation, and we are going to discuss some limitations that we have about that. Yeah, so the idea is to look for statistics tools that allow us to understand if we can justify that this common ancestor really exists. Okay, so let's check something here. For instance, I'm going to start talking about, not without a lot of formality, but this idea of statistical significance. This is what we want to check now. Yeah, so if you are checking for similarity, we would like at the same time, okay, we first start checking the similarity, and after the result that we get it, we will try to connect that with a number. And based on that number, we would like to see if we can trust or how much we can trust in that number that we are obtaining. Yeah, so we will elaborate some measures, or we will discuss some measures. The measure can be always computed, but not necessarily it means that because we can compute it, it means that will be significant. Okay, so this is one of the new concepts that maybe if you haven't take a statistics class, probably is not clear yet. Okay, so what happens when you find a statistically significant match? Yeah? So what we are going to try confidently is to infer that there are two sequences that are homologous. Okay, so when you are applying a tool of similarity and you find some matches or some results that are statistically significant, we are going to trust in that result and to try to infer that they are homologous. Yeah. Nevertheless, what happens if we don't find a statistical significant match? Okay, this sequence of database of sequences is always done using some database. Yeah, so this is one of the limitations that we have. It, of course, we are looking in some particular databases, and this is the information that we have there. Yeah, so of course, sometimes we can go and look for a database. Sometimes the information is not in the database yet. That doesn't mean that there is not outside, but there is no in the database yet. So this is one of the limitations that we have in this kind of analysis. Okay, so when you're considering this analysis of sequences and you are not getting a statistical significant match, what happened there? Well, unfortunately, we cannot be certain that no homologs are present. Okay, this is one of the things that we would like to highlight. Okay, how we check for statistical significance is very simple. Is by testing an hypothesis. And that's exactly the point behind using statistics. So we would like to check this technical word as main significance based on testing an hypothesis. Why is not working as a mathematical comparison? Because when you are considering samples, there is always some variability inside. Okay, so what it means, this variability, it means that you will always have errors. Yeah. So when you are looking for significance, it means that the error is not exactly as I mentioned previously, provoked by just chance. Okay. So our objective behind testing an hypothesis is just to minimize some errors. And which kind of errors we can find, but probably you know about that. Yeah, there are some false positives there. And what is the meaning of false positives? So is to get some non-homologs with significant scores. Yeah, so you are computing a score for similarity, and you get maybe that the two sequences are not homologous. Okay, so those are the errors that can happen sometimes. And yes, we would like to minimize those errors. Well, at least this is the first error in the context of hypothesis testing is type 1 error. And the type 2 error is when you have false negatives. What does it mean? It means that whatever you are computing again the score, and you are getting a score that is not significant, but at the end you have homologs, even if your score is not giving you significant results. Yeah, so this is the other error that we can find there. So in this hypothesis testing context, those are known as type 1 and type 2 errors. Okay. So in general, thinking in the tools that we are using here, if this is an introductory, you don't know what I'm talking about, don't worry, I'm going to address that later. So, but if you already run something like that, you know probably about BLAST, and we are now trying to understand Hammer. And these tools minimize false positive. Main limitation here, these tools doesn't make claims about the false negatives. Yeah. So we know and we understand that these tools, again, they minimize false positive, but we don't know anything about the false negative. Okay. So those are the limitations that we are working with. Okay. So how we address similarities, or how we assess similarities? If you have some couple of sequences, how do you test for similarities? Okay, so for instance, if you consider these two sequences, do you think that are they similar or not? So take a moment to check this one by one. So both start with C, both finish with D. It seems like they have the same length, same number of nucleotides in this case. So are they similar or not? What do you think? Well, if you don't know how to answer that, it's okay. What you can for sure recognize is that in these two sequences, you have 10 letters in each one, and you have two mismatches and 8 matches. Yeah, this is something that you can recognize. Uh, is that enough, or these counts of 8 and 2, is that enough to say that they are similar or not? We will see. That's exactly the point. So now let's try to check for this one. So these ones are using some amino acids instead. And you can see that now the sequences have a higher length. We have more letters here. What do you think? Are they similar or not? Well, if you check or you take a moment to see, there are some subgroup of letters that is repeated, but for sure, you can see that in the second sequence, there are some letters between the A and the blue one that I highlight here. Yeah, so the blue one is repeated, but in the second sequence, you can see that there are some letters in the middle between the A at the beginning. Okay, so what do you think? Are they similar or not? Yes, no, no, we don't know yet. Yeah. Well, maybe you know already, but this is the point of this first video to try to introduce the idea. Okay, so to quantify some similarity, sometimes it requires something that name is an alignment between sequences. And at the moment, we will assign a score based on the alignment. What I understand by alignment is something like that, where I can introduce some dashes here that the team can name maybe is a gap. And the gap represents some insertions of deletions. Yeah. What is the biological reason of that? Probably Dr. Perez can explain that better than me. Yeah, so you can ask him. Or you can ask some of your experience already, or maybe you know already, and I'm the one that have no clear explanation about that. Yeah. So, but this is the point of an alignment. So sometimes, as I mentioned, not necessarily the blue part that is the one that matches uh, are exactly like that, aligned. So we can introduce some gaps in one of the sequences to try to get more similarity. Yeah. And when you are analyzing something or you are proposing something like a score, this alignment is important to make the score to increase. Yeah. So, for instance, if you add this technical concept, so I mentioned what is a match, what it means that, of course, the letter is the same. A mismatch, it means that the letter is different. And a gap, it means that I introduce something like a dash, maybe here or maybe here. Yeah. And one measure of similarity in this case is very simple and very intuitive behind. We don't know how effective is this measure of similarity, but for sure is something clear, is to consider the number of matches divided by the matches plus the mid-matches. Yeah? And you multiply that by 100 and you get something like a percentage. It's very important to understand that this is after the alignment. Yeah. So in this measure of similarity, we are not including any information about the gaps. Yeah? So this is a simple number that we can compute it, and we can get some insights from this. But we don't know really how effective it is, in particular because we are not quantifying this uh, effect of adding some gaps in the alignment. Yep, it is clear. Hope it is clear. Mm. Okay, so when you're trying to elaborate alignments, you can have some global or local alignments. What does it mean that? In the global alignment, you're trying to align the full length of two sequences from the beginning to the end. Okay. So what is the limitation here? Is when you propose the alignment, you may have a lot of pieces or stretches of low similarity. This is one of the limitations. In the local alignment, you are just thinking in one subsequence of the sequence, as a part of the sequence. Yeah. And this is exactly the one that you want to align or make to match. Yeah. So in this particular example, maybe you can have these two sequences, sequence 1 and 2. You may see from the beginning that they don't have the same length. Okay. So when you elaborate the global alignment, what you can see, okay, in this particular case, you are considering the full length, and you would like to align uh, both sequences entirely, and you are introducing a couple of gaps in the second sequence, yeah, in order to get some matches here, in particular the A's, the C, the G. Yeah. This vertical line is that what it means that you have matches. Yeah. In the local alignment, you just focus in one particular part of the sequence, yeah, and this is the one that you want to align. Yeah. So in this particular case, you are using the information in the sequence 1 from the third letter to the sixth letter. Yeah, yeah, something like that. Seventh letter. Okay, and you don't care by having an alignment that consider the full sequence in the case of the second sequence. Okay. That does make sense? Hope it makes sense. Yeah. Otherwise, I can get more details in another video. This is an image that I took from this particular reference. Is a review on sequence alignment algorithms for short reads based on next generation sequencing. This is the figure 2. So this is some description of the figure and what is the meaning of each one. For instance, there are dots, long lines, and short lines. So no matching pairs are indicated with dots. And this is clear here. Yeah. These dots, that's what is the meaning. C against A, okay. It means that they are non-matching pairs. T against C, A against C. Okay. Matching pairs are indicated with long vertical lines, are the one that I mentioned previously, and insertion or deletions, which show blank spaces, are indicated with short horizontal dashes, yeah, as the gap that I mentioned previously. Okay. So this is an image that I took from that reference, in case that you are curious where I took it. Yeah. Okay. So when you are trying to reach something like a number to see if the sequence are similar or not. As the one that we did previously, this is something like a very basic measure of similarity. Yeah. And I mentioned previously, it doesn't consider, for instance, the effect of the gap. Yeah. So neither it consider the meaning of the letters. So one alternative here is to consider something like a measure that includes that importance. So sometimes we can find something that the name is a matrix of similarity, and that provides you some scores associated with meaning, in this case, of the letters. In this case, I'm showing you just a piece of a matrix that is used for understand an amino acid sequence. Yeah. So you may see here, uh, let's see, you can see that in this matrix you can have uh, new values, for instance. New values. Yeah. What does it mean? It means that when you find in this case an R with a G, with a T or with a V, it means that these matches or these mismatches, I'm sorry, these mismatches won't affect the score in this case, yeah, because the value is zero. Yeah. In the case of for instance, the diagonal that you can see here, you are seeing that R is matches R, or G is matching J, I is matching I. Yeah. This one is like the identity diagonal. Yeah. Those are positive numbers, and you can see they are different. And why they are different? Because we understand that each amino acid has a different importance. That's why. Yeah, but all are positive. So in this case, R matches R. In this case, it means that you have identities. An identity. Not just a match, but an identity. And again, there are different numbers, all are positive, but there are different numbers here because again, there are some amino acids that are more important than others. In the same matrix, you can find also some other positive values like this one or like this one. Yeah. Not necessarily it means that when you have an identity, is the only case that when you get a positive value. Yeah, so these particular cases, for instance, when you get an I and a V, you can see that you are going to add or increase the score in this case. And finally, you get the negative ones, and the negative ones, what it tells you is that you are losing similarity in some sense, like this one, or this one, it means that you are losing similarities. Yeah. So trying to understand this, it is this description here. Identities and conservative replacements has positive scores, while unlikely replacements have negative scores. So the R and the F, we understand that as an unlikely replacement. Yeah. So this is something that is decreasing the similarity in when you are comparing the two sequences. But again, they may have some conservative replacements, like this one, the I and the V, that it provides you 3 points to the score. Okay, what does it mean? It means that this is something that can happen, and even if they are not matches, they add some value to the similarity. Okay, this is one matrix that is associated with the analysis of a protein sequence. Yeah, you have amino acids. This is just a portion, just a piece of the matrix. Why? Because if you remember, you have 20 amino acids. So the real matrix, you have 20 letters here and 20 here. Okay, so it's a very, very big matrix. Okay. Questions about that? No? I don't know if that's clear or not, but if you don't have questions, maybe it's clear. No, no. The point is that, yeah, so try to provide me some feedback to see if there are some things that are not clear. In case that is clear and it's very simple, I will continue. So our next step will be to understand how do we get the sequences, to try to start analyzing this. And this is actually the point where I'm going to stop this recording. Instead of doing the next step and go to see and to understand how we get some sequences, I'm going to stop here and again sharing this short video to you to get the first step in the context. Yeah. Hope it is clear, and it is not clear, please let me know. In that way, I can start try to clarify some terms in the second video. Thank you very much.